Hi everyone, thanks so much for calling in. My name's Amy Willis and I'm an assistant professor at the University of Washington, and it's my very great pleasure to be giving a presentation in the Northwest Data Science Summit, and I really want to thank the organizers for putting this on. So I'm going to be talking about the microbiome and ways that we commonly measure aspects of the microbiome, and ways that we commonly model aspects of the microbiome, and maybe some recent research in that area. So in case you don't know, accompanying you with you throughout your whole life is your microbiome. So your microbiome is the complement of all microscopic organisms and the genes that they carry and the metabolites that they produce. Your bacteria are some of the most diverse life forms in existence, and in fact we have to thank them for our existence because bacteria are some of the earliest life forms, and they oxygenated the atmosphere for us. They play critical roles in nutrient cycling all over the world. So carbon cycling, nitrogen cycling. They do bioremediation. They break down nuclear waste and other biohazards. Your resident bacteria help you digest nutrients from your food. They fight off bad bacteria, other pathogens. They fight off viruses. And in fact, all eukaryotic life descended from bacterial origins, so they're not so much your distant cousins as much as your distant ancestors. So how do we go about studying the microbiome, or the complement? In this case, I'm going to talk about bacteria, the complement of bacteria that are present in an environment. So we can't go out and do an ecosystem survey and count the number of them, like we would count the number of tigers and elephants in a traditional macroecological survey. Because they're too tiny, and also they're just too numerous to study sort of directly. And so we study them indirectly through incredible technology that sort of is incredible set of technologies that collectively is known as high throughput sequencing. And so I've got this very simplistic typewriter here. Commonly for studying human microbiomes, we'll sequence human fecal matter, which is a snapshot or an approximation or a snapshot of your gut microbiome previously. Essentially, we'll smash up all of those bacteria, we'll put them in this very fancy blender, break open the cells, look at the DNA that's in there. We'll use a fancy typewriter machine to sort of figure out all of the DNA complement that's there after it's sheared up into pieces. And then we'll reconstruct from those sequences who was there, essentially the DNA signature of different microbes that were present in the original environment. And so then what we can do is count them up. And so at the end of a very sophisticated set of wet lab pipeline, and at the end of a very sophisticated bioinformatics or post-processing pipeline, we end up with essentially a count table. So I'm going to use WIJ to refer to the number of times that we observed in sample I, strain or microbe or bacterial strain J. And so, for example, in sample 1-1, we observed Lactobacillus crispatus 13,670 times. And so what I'm going to be talking about is how we might model this data. And if you're familiar with microbiome data, with different technologies, we might have slight variance on a table like this. We might be looking at coverage, so average number of reads recruited across the genome of a bacterium. But we can sort of, for the purposes of this talk, I'm going to be focusing on count tables like this. And so the way that this technology works, the sum over a sample, so if we sum across the columns for a given row, we can total up the number of bacteria that we counted in that sample, and that number is a random number. And so it's not necessarily informative for how many bacteria are present in the environment that the sample was taken from. And so what we're going to do, what we can do is compare counts within samples, but not necessarily across samples. And so the way that this field has proceeded over the last 15 years or so is to consider a very sophisticated set of models, maybe negative binomial models or beta binomial models or zero-inflated negative binomial models. But underpinning all of these assumptions is this underpinning all of these models is this fundamental assumption that the relative abundance of a microbial strain within a sample is in expectation a unbiased estimate of the true relative abundance of that strain in that sample. So if you're a statistician, you can think of this as the observed relative abundance is approximately the true relative abundance on average. And if you're a statistician, you're comfortable with expectations and probability, I'll be talking about the expected value of the sample relative abundance as being equal to Pij. So this is an assumption that underpins lots of different types of models, including DESeq or LEFC, which is a microbiome differential abundance testing method, corn-cob, and tons of others out there in the statistical and the computational biology literature. An equivalent way of thinking about the same thing is to think about the observed count as being approximately equal to the true relative abundance multiplied by some sampling intensity. So some samples are just randomly sequenced more deeply than others. And so we can think of this as the expected value of those counts is equal to something true that we care about in the environment, and then something that's consistent across all taxa for a given sample. So these two slides are saying the same thing. So that's what's been going on over the last 15 years in the sort of computational and statistical world. But in the lab, in the way that folks have been studying the microbiome, folks have been thinking about it slightly differently. And so what we can see here is the relative abundance at a very broad grouping of bacteria of an identical sample that was sent to about 12 different labs, here. So just want to be really clear, this is an identical sample viewed through the lenses of 12 different laboratories who perform this sort of sequencing. And what you can see is some pretty staggering differences in not only how much of each bacterium is there, but even who is there. And so what we're seeing is that different labs are getting different estimates of the relative abundance of different taxa. So this idea is incompatible, let's say, with the model that I presented earlier, where the expected relative abundance that a particular lab sees as an unbiased estimate of the truth, because essentially they can't all be right. They can't all be unbiased for the true abundance of this particular sample. Okay. So how can we think about this? So if you ask the lab folks, they say, well, there's lots of different ways to do high throughput sequencing. There's different ways of storing the samples. You could freeze and thaw them in different ways. You could break the cells open in different ways. You could choose to do amplification sequencing. The way that you do bioinformatics or post-processing can matter. And there's a very rich literature that shows that every single one of these decisions that you make at all of the steps in this wet lab process influence the resultant tables that you see at the end of the day. So I would say this is not a great thing to learn as a modeler or a statistician, or I guess for anyone to really think about, because then we sort of get into these arguments over, well, whose is better and how do we evaluate whose is better? Are people even seeing the same thing? Is anyone else's data reliable? What's the value of public data? And so on. So there's lots of reasons that we want to sort of reconcile these views. And so a lot of my work in the last year or two has been trying to come up with a modeling framework that describes these two key phenomena that I just described, or two key ideas maybe, that reflect both the statisticians and the computational biologists' view of this data, and also the way that it's generated in the lab, and our understanding of what different labs are seeing. So the first aspect that we want to capture, that we want to investigate, is the idea that this data is informative for the relative abundances of microbial strains in samples. And the other idea is that everyone's data produces some different estimate of relative abundances, even if they're given the same samples. Okay, so what I started doing with a couple of terrific collaborators is saying, well, how can we sort of get back to basics and think about what this data is showing us? And the way that I'm going to do that is to look at a couple of different, or I'll show today one data set where we compare the results of sequencing and the profiles that result from sequencing to what we know to be present in the environment. So that's what I'm going to talk about now. So this work is joint work with Ben Callahan and Michael McLaren, who who are both at NC State and are just terrific collaborators. I've been very lucky to work with them. Most of the ideas that I'm going to present today are summarized in this paper that was published in eLive last year, so feel free to check it out. Okay, so how are we going to evaluate sort of the quality of this data? We're going to look at what's called mock communities. So mock communities are artificial microbiomes. They're essentially constructed communities where we know what's present in those environments because they've been essentially constructed in the lab, and they're commonly used for different benchmarking purposes in protocol development for microbiome studies. And so this data set's published by Brooks and co-authors in 2015. It's a very rich data set. What we see here is a mixture of 8 different samples containing 7 different bacteria, which are represented by the different colors. And we see that all of these bacteria have been mixed in 1-to-1 ratios with some combination of the bacteria that are present were possible to sample from. So in the first sample, we've got 1-to-1-to-1 ratio of S. amni, S. agalactiae, and A. vaginae. These are all human vaginal associated microbes. And so if we had a sequencing technology that could perfectly reflect what was present in the community and was an accurate estimate of the actual abundances of taxa in this community, our observed profiles would be identical to this true one. That's what an unbiased and zero variance technology would give us. Alas, this is what we see, which is not so great. So we see a couple of taxa are consistently over-observed, we see a couple of taxa that are consistently under-observed, a couple where it sort of could be either way in some samples and not others, but overall not a particularly flattering view of what this technology is producing. And for the microbiome folks out there, I'll say that you can do the same thing for copy number adjusted, 16S copy number adjusted sequencing of the observed profiles, and basically doesn't look any better. Feel free to check out the manuscript for more details. Just in case you weren't convinced from those last plots that we were sort of all over the place in terms of our correlation between our true relative abundances and the observed relative abundances of the taxa, we can see a scatter plot here, equally not as flattering. And again, it sort of looks all over the place. There's some taxa that are consistently over, some under, and some that are sometimes over and sometimes under. So what hope is there? Okay, so what I'm showing here is our attempt to look at is an analysis that we did that's described in the paper where we tried to say, well, if relative abundances are all over the place, are there any features of these communities that are more consistent? And so here what you'll notice is that these 5 communities all have this green taxon and this blue taxon in all of them. And so what we see, and we know that for every one of these true relative abundance profiles, we have a 1-to-1 match of these taxa. And what we see is that the green taxa is consistently over-observed, and exactly how over-observed it is varies sample to sample. But if we look at the ratio of this green taxon, which is Lactobacillus iners, to the ratio of S. agalactiae, which is in blue, we see a pretty consistent factor by which Lactobacillus iners is over-observed. And essentially we see that it's consistently about 18 times over-observed compared to S. agalactiae. And so, what can this tell us about the way that we should be analyzing microbiome data? And actually, before I say that, I'll say this is not a phenomenon that is unique to these 2 taxa. This is actually a consistent ratio of observed to expected in these 1-to-1 mixtures is observed across all pairs of taxa in this data set. So that's encouraging. This is telling us that even if we can't get good unbiased estimates of true relative abundance, we can get perhaps an unbiased estimator of some function of the relative abundances, for example, their ratios. Okay, and so what does this data potentially tell us about the way that microbiome observed profiles are reflective of what's actually present in the environment? So I'd say this data suggests the following model. It's telling us that the observed relative abundances are proportional to the true relative abundances that are actually present in the environment multiplied by taxon-specific efficiencies. And so not only do we have some sampling intensity factor, which is essentially given by the denominator in these equations, but we have some factor by which different strains are consistently over or under observed relative to all of the others, which is kind of fundamentally different from the previous model where we just said observed relative abundances are in expectation, or essentially unbiased for our true relative abundances. So this is can actually have some pretty profound impacts on the way that we can be analyzing microbiome data. So just to give a quick run through of what I'm talking about when I say that, if we look at this observed profile, we see that it looks like the orange taxon is less abundant in the group on the left, the treatment group, let's say, compared to the control group on the right. But in fact, it actually could have been less abundant in the control group compared to the actual abundances if we had taxon-specific efficiencies that looked like this, where essentially we had this green taxon, which is changing in abundance between the treatment and control, is a very high efficiency taxon and sort of eats up the slice of the pie that could be that, and takes it away essentially from the orange taxon. So what we're seeing is that we cannot accurately estimate changes in proportions, and actually we don't even correctly infer them in the right direction under this model. That said, if we looked instead of the ratios of these taxa, we could actually get a sense of how much these ratios are changing by, and so we could actually estimate changes in ratios. So that's a big shift in terms of what we can estimate based on this model. And so these results are described in this paper. If you're interested in more details, feel free to check it out. The way that we're proposing to go about sort of calibrating these models is with essentially a different data generating process, or a different model for the data that we observe that does reflect these efficiencies. And I was fortunate also to be involved in a collaboration with Brian Williamson and Jim Hughes, both at UW, who were to construct a model for essentially calibrating to estimate the true relative abundances based on observed relative abundances, and perhaps some low throughput but high quality data that we could use for calibration. Okay, so I think there's a couple of really important implications here for statisticians and for data scientists. And I'll let folks who are more interested in the microbiome side of things take a look at the paper. But essentially, over 10 or 15 years, there was a lot of work in the computational biology and statistical literature proposing estimators and hypothesis tests that were based on models that assumed that the true relative abundances could be unbiasedly estimated by these sample relative abundances. And the data that I showed today show that these are first-order misspecified models. Essentially, we do have, those are biased estimates in the presence of unequal efficiencies across taxa. And so what we did in this paper that was published last year is essentially evaluated that assumption. And this is an assumption that's been used for maybe 15 years in this literature. Model evaluation happened really, really late here. And so it calls into question a lot of the findings that have been made based on these models that were previously proposed. And so that's an ongoing process and something that my lab's involved with. So I would say sort of the shortcoming here is that very few folks in these communities, in the statistical community and in the microbiome community, speak both languages, one of statistics, of biology, or maybe statistics and biotech, sort of the one that allows us to evaluate this technology in the way that it's being used. So if we're looking at this technology and thinking that it's producing estimates of relative abundances, really that should have been something that we evaluated really early. And so what I would really love to see, this is what I hope the take-home message for everyone will be, is that interdisciplinary collaboration is challenging. It takes a lot of willingness to learn, willingness to sort of learn a new field, learn a new language, but it really is the only way that I think we'll move forward. And so with that, I would love to thank my collaborators, work with so many fantastic folks on this project, and also to thank the organizers of the Northwest Data Science Summit. Thanks so much, and I'd be happy to take questions during the Q&A session that we should have in a few minutes. Thanks so much.